Let me go ahead and introduce you quickly. So Joel Karpiak is the head of Protein Design and Informatics at GSK. Focusing on, I believe, computational chemistry and protein design. Worked a lot in drug discovery, done quite a bit with GPCRs, which is really cool. And I think is now working probably with integrating artificial intelligence for drug discovery and industrial applications, maybe. But anyway, take it away.
Thanks. Yeah, a wide variety of things. So leading the Data and Predictive Sciences organization has been interesting recently. So I do want to talk about, because there's such a wide variety of things, especially over this workshop, how we all can work together as a team for translation, and reflecting on what translation means in a variety of different contexts is kind of important, I think, for actually impacting how we accelerate or make drug discovery a bit more efficient. So just being able to talk to each other and sharing the same kind of vocabulary around what a data scientist does, the engineering needed for an actual compute cluster, what a computational scientist needs to do for actual molecular design, or wet lab scientists able to actually test it. How do we contribute to each other's disciplines? How do we impact and communicate what we're actually trying to predict, get those experiments done, and influence each other to really work more efficiently? Even something very simple in terms of how does DNA correlate to RNA, which translates into proteins. You know, people who talk about genes as targets are very different than proteins as targets. How do we all speak the same language there for a common goal? And then also translation in terms of a protein target, but then finding a binder to that is very different than being efficacious in a model system, and certainly different than efficacious in the human system. And so how do we line up modeling to be able to impact all of these things in an efficient way? And I should say that this slide is really messy on purpose, not just because I have poor PowerPoint skills, but because it is a really messy area that I don't think anybody quite has right. And although computers, obviously, as we've heard throughout this entire session, can speak all of these different languages, we don't yet have completely autonomous agents doing everything possible. Humans are still in the loop, still need to make decisions, and still need to make prioritizations. And how do we do that in an efficient way? And so one thing that we try to do is really start out, when we start with a project, have informaticians at the table to really map out what a data strategy would look like continuously throughout target discovery, the actual medicine, and through development. So we don't have a lot of this handovers, knowledge gaps, and also any kind of white space, because we're doing things in a modular way. But really trying to have that seamless data flow so that we're generating data, making predictions that impact really local and next decisions, but looking at it from a global mindset or a foundational mindset and building up those data sets as we've talked about previously throughout this workshop. And really, this is a seamless integration between the discovery platforms that might be a bit bespoke, whether there's a chemistry or biology, data scientists who enable the analysis of the data, and then the automation to be able to do it at scale, and the quality that we need to be able to make predictions. And all of this has underpinnings of transparency and trust within the team so that we know what's going on, people understand where predictions are coming from and how to interpret them in the best way to make the best kinds of decisions. And so we've heard a lot about different kinds of machine learning and drug discovery and things like this. There's a wide variety of things where, especially machine learning and computational methods, can have a huge impact. So I just want to focus a bit on what happens in small molecule design, or can happen in small molecule design to put all of these things together. So that's around generating data. How do we do it intentionally? How do we incorporate it into models, and then also deploy those in a responsible way for people to be able to make good decisions in a bit of a sandbox environment, and interpret what we're actually trying to say from a computational lens into the actual experimental practice. And so a lot of this goes into a bit of a framework that, at least at GSK, everybody has their own platform. Here it's called Bradshaw for our automated small molecule design platform. And I should say, for starting with this and for the rest of the talk, in principle, there's really nothing different between small molecules and large molecules or oligos or anything else that we might be interested in designing. Just happened to really select this to get into a bit of the nitty-gritty about how teams work. And so here, we've got sort of all the components that we've heard about already throughout these sessions in terms of molecule generators, model collections that we call QSAR Studio, that have a variety of different amount of data behind them to enable a quality model building, and then also selection or ultimately prioritization of molecules to make. And how do we do that both on automated chemical platforms, array-based chemistry, bespoke syntheses, in a way that really drives the program forward and with maximal amount of impact and information generated at each stage. And so I just want to kind of give a couple of examples around what this might look like. And so the variety of different types of data are going to be important for the kinds of models that we need to build, both on-target, off-target potency, DMPK models, and how do we deploy these in a responsible way, again, to make decisions. So the data could be images, it could be assay readouts, it really could be anything. And so being able to have a really flexible system for automated model building and deployment is pretty important, obviously. And then sort of standardization about how we build those models, statistically test them, and then being able to select the best ones that are generalizable going forward. And so I just wanted to make another comparison to a bit. We've heard a lot about open data versus proprietary data and things like this. Being able to combine them in a statistically rigorous way is pretty important. Unsurprisingly, our own company's data is biased towards inactivity from a variety of screening cascades, which is probably similar across the industry. But what gets published is typically pretty positive. So being able to actually establish, through carefully curation, establish balanced data sets for model building is pretty important. And again, I want to emphasize the importance of being able to incorporate that open source data going forward. And so just some things to take into consideration, again, because this is a theme, in terms of evaluating experimental data for model building. This requires really close collaboration and communication with the experimental scientists to make sure you're interpreting the experimental data in order to build the best kinds of models. So within the actual experimental assays, there could be noisy measurements, which impact things, the actual error prediction, completely imbalanced data set, depending on whatever the results are. The fact that you're simplifying and having reduced representations can have an impact on how you actually build the model and the ultimate quality of it. The ability of the model to extrapolate based on the data in the training set, the ultimate uncertainty within the model and how we quantify that. And then also build in interpretive methods, again, for experts and potential non-experts to be able to make decisions off of and understand what's actually going on under the hood. And then over time, unsurprisingly, as chemistry changes, the program evolves, the ability for any kind of the series or scaffolds or functional groups over time, they're going to contribute very differently. And so there's a dependency on a lot of these kinds of things in order to generalize. Missing data sets, sparse matrices, tons of problems that you have to take into consideration when we're looking at it in order to do this in a rigorous way. And so being able to then, both at a global level, build data sets intentionally to be able to build the best kind of models that extrapolate across chemical space, while at the same time understanding the limitations on a potential series that you're working on, and then the need to build a local model in order to fill in space and also contribute to the actual project that you're working on in order to drive it. So understanding whether or not those kinds of global models are suitable, if you need to build a local model in order to push it a bit more, or if you can generate data in order to make both better at the same time. And what are the strategies in order to actually do that? I should say, just to give a sense of scale about the kind of data and the kind of models that are necessary for driving these kinds of projects forward, just to give an example here for our off-target assay panel, lots of nasty things that you want to avoid for various reasons and downstream effects. Here we're showing a sampling of 98 models based on a panel of off-targets that's pretty standard, and showing different levels of model quality based on the data that exists and whether or not we can actually improve that with intentional data generation going forward. So again, just to give a sense of scale of what needs to be done in order to design a really good molecule, and the sort of data gaps that we have in around that. And getting back to sort of teamwork and collaboration, talked about working with the experimentalists in order to interpret the data correctly for model building. Now it's about interpreting model building correctly and deploying those models for experimentalists or chemists or whomever on the other side to be able to actually use appropriately under supervision in sandbox, come up with their own ideas, do design, and how do we pool all of those ideas together to make the best decisions. So one potential way to do this is through a system, for instance, this is a commercial one with Road called Live Design, where it's a cloud-based system, a cloud-based platform for design that's accessible. You can deploy models through it, get immediate feedback on different kinds of design ideas, and incorporate anything else that you want to in order to actually push these kinds of projects along. So for instance, you can build custom templates for users to be able to look at data in a certain way, again, as the sandbox to make sure it's being interpreted in a certain way, and incorporate all of those other kind of model endpoints in here that might be important. So not just the prediction, but the uncertainty related to it, how it should actually be interpreted, and the appropriate visualizations if necessary in order to actually push things along. And depending on the things you might be interested in, there could be a particular view or template for safety, for off-target effects, for on-target potency, anything you really might be interested in can be deployed here. And so this is really a way to have transparency, especially with experimentalists and computationalists, about how models are doing, how they're being deployed, and how they should be interpreted to make the next decision and design a test cycle. And again, just to give a sense of scale, so on each project, we've got over 150 global models that are continuously rebuilt that are various different properties that would be interesting or important for actually driving those projects forward in that particular series. And then, unsurprisingly, many custom models depending on the actual series or the target specific that can be deployed through here. And this is always a collaborative effort, for instance, with DMPK or other kinds of assay scientists in order to be able to do this. So again, many cooks in the kitchen in order to try to do this. But the close collaboration between the different functions, the transparency and the trust there is absolutely essential from the beginning. And so try to do that both on the experimental and on the computational side. Again, another aspect of transparency is the automated model drift. So for these types of global assays or global properties that we might be interested in, anybody at any time can look at how that model is performing with an automated system flagging if it starts to perform more poorly as more compounds get made, and suggest that the model actually needs to be remade, or we can consider an active learning strategy to actually make it better. But again, in the interest of transparency, these are things that anybody can look at any one time, even though there's an automated system to flag issues. Just to ground it in reality a bit, for instance, one project, just to give an example of how this works on individual properties, and then also how it could work in combination. So here, there is an off-target liability here for a particular series on a project. And in order to try to get around it, we had focused data generation based on the model that the global one is not translating. And so being able to decrease that over time through these different kinds of active learning iterations was pretty key to the project. And at the same time, providing the kinds of visualizations and justification for why we needed to do this. So for instance, on the right, showing that all of the global model predictions were clustered in a different kind of chemical space that were not applicable to the local things that we're actually interested in. You can see in the first two columns. The chemistries are very different. So things like this are pretty essential for communication about prioritization, experiments that need to be done, as opposed to just saying it or taking somebody's word for it. It's an essential part of sort of the project team moving forward. And so looking at this as more than an individual property, so looking back at that off-target that I mentioned before versus on-target potency, being able to decrease or tune out the activity that we don't want while keeping the potency on the actual on-target at the same time. So using that example as just on and off targets is a pretty good proof of concept, and getting back to how we actually do this in a practical way. Working on multiple series at a time, for instance, we really try to map out, getting back to that data strategy and being intentional about what we're doing, map out how long these DMTA cycles actually take based on the chemistry we're interested in, and stagger the chemistry resource with the screening resource with the computational resource all at the same time, so that this process can be as efficient as possible while also generating the maximum amount of impactful data that we can do at the same time. And so this is pretty much essential when maintaining a large portfolio of projects, as opposed to a startup or something really focused on one target and putting all the effort and resource behind it. And so I should say too, then you could do this at a larger scale about different kinds of properties that you might be interested in. So when starting a project, there's, unsurprisingly, you would want a soluble compound, or you would want something that's permeable for various reasons. And there are general guidelines for each one of these things. And so for an oral drug, for instance, and so an easy way to do that is to just take general guidelines about what's good there, try to design compounds against them, whether using filters or actual multi-property optimization techniques, and do those over time. That's great, right? We can do that, and this shows an example about different kinds of properties that have become important based on the series and flags over a variety of iterations and over time on a particular project. But unsurprisingly, what this does is it potentially gives you good-ish compounds in general, but there might be something specific for your particular target, for your particular disease, or for your particular mechanism of action that is important. So understanding what a good molecule or a candidate molecule in that particular thing is as early as possible is really important to save you from making hundreds of molecules, for instance, that you don't have, and realize how good everything that you actually have is. It also helps you set up objectives for multi-objective optimization at the same time that are specific to your disease or even your series or modality at the same time. And so one potential way to do that is actually taking a concept from clinical modeling, going back to translation, which is these coarse mechanistic models known as QSP or PD modeling, so quantitative systems pharmacology. Build these for each one of the diseases where you know, for instance, by the time you have a candidate, how these things actually perform, or how these molecules actually perform in vivo, or maybe you even have clinical data from a disease that you've been investing in over time or have bought that data, for instance. And then also understand the PBPK properties of a particular molecule or that series. And so working backwards from those, so going backwards from the clinic, so a bit of a reverse translation approach, you can predict all of these ADME properties theoretically with really good models. So understanding how the individual components of permeability, solubility, clearance, all these things that bioavailability, all these things that are important individually, how they combine into actual PK properties, and that then translate into PD properties to be efficacious in human. So being able to then take theoretically these models, being able to predict the PK/PD profiles of these molecules that we're interested in, and then again work backwards. So being able to virtually profile hundreds of thousands of potential molecules, understand the drivers of efficacy that are contributing to the PK. So that could be solubility, that could be permeability, or it could be a really complicated contribution of all of them into once. But using different kinds of machine learning to really interpret what's going on there, to identify what the design strategy should be going forward. And then for your next iteration, you can enumerate all the different kinds of virtual compounds for the series you're interested in, build better models, and then keep iterating through the series. So it's not just playing whack-a-mole and saying, oh, I really want to optimize solubility, I really want to optimize permeability, but thinking again globally in a really large translation point of view, how do we care about human dose ultimately as a thing that we're trying to optimize as early as possible? Just to give an example of what this looked like, you could have two different compounds, so labeled A and B here, where one is less potent than the other one, but different balance of ADME properties versus potency. And so these kinds of visualizations about the probability of what the human dose might end up looking like kind of makes it clear about what the design strategy should be and which series maybe we want to prioritize with more resource going forward. So do we think it's easier to optimize potency but keep ADME the same? If we reduce it by 5x, we can increase the probability of a lower dose much more. Or is it going to be harder to actually optimize the ADME properties, for instance? And then we can use interpretable machine learning techniques to really break down which physical properties contributing to the overall ADME score to optimize for further design rounds. And I should say too that there are other kinds of properties that impact that kind of assumption, where there might be variables or biological variables we don't understand, particularly if it's a new disease or a new project or things like this. So we can make a series of assumptions and understand how those look over time. So for instance, the contour plots on the right are different levels of efflux ratio in the same plot that I was showing on the previous slide. And so being able to show how that changes over time and the probability of success really might call out an important parameter, for instance, that we need to prioritize for experimental testing to really understand what our compounds are doing or whether or not a series is worth pursuing. So it highlights basically the things we don't know and should help prioritize the resource around certain experiments that are clear go, no-goes for the project or for the particular series. Same sort of thing just around turnover rate of the actual target and in a PD sense. And so you could have the same molecules in terms of potency, ADME properties and things like this, but depending on what the actual properties of the molecule and the target are, it could completely have a 0% chance of success or clinical efficacious response rate, or it could have a 50% chance of success. And so being able to highlight if these are the proper experiments to be able to do to interpret, to tell you what to do next is important for prioritizing resource. And so going back to compound design, where this could come into play, especially as early as possible or where you want it to, is taking initial HCS hits. So for here in the middle is that is that black box. Doing different kinds of molecule generators or virtual enumerations around those scaffolds, different kinds of analogs, virtual screens, things like this, to understand the optimizability or the potential of that series to reach any kind of probability of success of being efficacious or with a low dose. And so being able to then take those series, look at the common properties of any of those molecules that are predicted to have a lower dose, understand where those key pharmacophores are, and utilizing them for further molecular design. Or if those series, no matter what we do, how much design efforts we put into it, is never going to be predicted lower than that, then either we can kill the series, we can reprioritize, we can test one or two key things that are the virtual nail in the coffin to make sure we actually have confidence in the model predictions. It just informs whatever the next decision is for the design strategy. So yeah, just trying to think again globally how modeling, different types of modeling translate all the way from the very beginning of hit ID to the clinic and feedback from that to answer questions about how do we actually modulate this target in an efficacious way, what modality you want to choose. So for instance, covalency versus protax degradation versus actual functional inhibition. And then prioritizing different kinds of compounds. What are the actual gaps to build a better model that we might need or to make more accurate predictions and prioritize those experiments? And then again, what is actually driving the biological efficacy and what are the physical properties that correspond to that are clear and interpretable for us to design to? So with that, just want to say thank you to everybody. And way too many people at GSK and elsewhere to thank. So this is just a very general one. I appreciate it. And hopefully that got us on time.
That was perfect. That was perfect.
Right.
That was great. Thank you very much.
Thanks.